Thursday's 2 o'clock Group 2 results of the dihybrid cross. Throughout the past 12 weeks, the anthocyanin and Brassica rapa plants and the color of the leaves are what was used in the experiment to prove if Mendel's law of inheritance applied to the experiment. Anthocyanin is the pigment which would result in a purple stem. No presence of the pigment would result in a green stem. The P generation started with a homozygous dominant parent crossed with a homozygous recessive parent. The homozygous dominant parents were purple stem plants with green leaves, and the homozygous recessive parents were the green stem plants with yellow leaves. These two parents were a true breeding cross and formed an F1 generation, which was a dihybrid cross. A dihybrid cross is a cross of two parents which are heterozygous for each trait. For the lab, the F2 generation was mainly the focus of the experiment, which was to see if the results from Mendel's law were reproducible.
In the beginning, a quadrant was obtained, and each cell contained soil, 3 fertilizer pellets, and 2 to 3 seeds of the seed sample number 2, with a wick that extended out from the bottom, and then the plants were watered. From then on, the plants were placed under lights that were 2 to 3 inches above the plants and kept under those lights 24 hours a day. The plants were checked throughout the week, making sure the soil didn't dry out. After 4 to 5 days, the phenotypes were recorded of all plants that grew. The top 4 healthiest plants were chosen in each cell to keep, and the rest was thinned out. Next, over the course of 14 days, the flowers were pollinated. A bee stick transferred pollen from one plant to another. Then after 2 to 3 days, the pollination process was repeated a total of 3 times. After the third pollination, the unopened buds were pinched, and the same process was continued for the next 2 weeks. The seeds were then harvested after 21 days of the pollination date. The plants were dried out and pinched to collect the pods to germinate. F2 generation plants were grown the same way as F1 generation plants. The quad had 4 seeds in each cell, totaling 16. Then the seeds were observed after 3 days, and the phenotypes were recorded.
None of the plants resulted with green stems and yellow-green leaves. There was a total of 9 purple stems with green leaves, 2 purple stems with yellow-green leaves, and 6 green stems with green leaves. The genetic explanation. According to Mendel's laws, the expected offspring of a dihybrid cross should follow the phenotypic ratio of 9:3:3:1. To illustrate this, the expected ratio of the purple stem and green leaf phenotypes should be the most prominent and should result in a 9:16 ratio because purple stem green leaves plants were the 2 dominant alleles for each trait. This would either have to be homozygous dominant or heterozygous for each trait to express dominance.
Continuing the expected ratios, the 3 to 6 ratios apply to purple stem yellow leaf plants and green stem green leaf plants. The purple stem yellow leaf plants had a genotype of either heterozygous or homozygous dominant for the presence of anthocyanin and a homozygous recessive genotype for the yellow leaves. The green stem green leaf plants had a genotype of homozygous recessive for green stems and a homozygous or heterozygous dominant genotype for green leaves. Lastly, the 1 to 6 ratio applied to green stem yellow leaf plants. The green stem yellow leaf plants had a genotype of homozygous recessive for both the green stem and the yellow leaves.
In order to test whether Mendel's laws of inheritance were present in the experiment, a chi-square test was done for individual group data and class data. A chi-square test is used to test whether there is a difference between the observed and expected values of the experiment. The lab calls for the observance of 4 different phenotypes. To determine the probability value, you must find the degrees of freedom and have a specific p-value, which can be used to determine the significance of the data. The degrees of freedom for the dihybrid cross was 3, and the p-value was 0.05, which resulted in 95% confidence. This all corresponds to a probability value of 7.82. In order to follow Mendel's laws, the chi-square value that is calculated must be lower than the probability value. The group and class data's chi-square value ended up being less than the probability value. This means that the null hypothesis cannot be rejected and the data gathered matches Mendel's laws.